Hello and welcome to Cabruin Aquarium's Discovery Lecture Series. I'm Julie Passarelli, the Education and Collections Curator at Cabrillo Marine Aquarium. Cabrillo Marine Aquarium is owned and operated by the City of Los Angeles Department of Recreation and Parks, and we're extremely grateful for the City of Los Angeles' support. I would also like to thank the Aquarium Director, Chrislyn McCarran, and Jim DePompe, the Programs Director at Cabrillo Marine Aquarium, for all their support. And a huge thank you to everybody who's joined us tonight. The aquarium remains closed due to COVID-19, but please check back on our website, cabrillomarinaquarium.org, for any new information about opening dates or any of our online programs. Before we get started, I would also like to thank and acknowledge the Friends of Cabrillo Marine Aquarium for their support. We plan to continue this lecture series through the rest of the year and spring 2021 online. The upcoming lecture is on Friday, December 4th, and Dr. Jay Smith from Cal Poly Pomona will be joining us. And the title of his talk is Rocky Intertidal Ecosystems in Urbanized Southern California, Long-Term Change, Human Impacts, and Management. We are currently planning the schedule and speakers for 2021, and they will be posted on our website soon. But again, all spring 2021 lectures will be online. If you're interested in any of these lectures, mark your calendars and check our website for details. You will need to RSVP like you did for this lecture on our Eventbrite to receive the webinar link. So now it is my pleasure and honor to introduce tonight's speaker, Dr. Nicole Bonuso. Dr. Bonuso is an associate professor at California State University, Fullerton, specializing in invertebrate paleontology. She did her grad work in paleontology at Syracuse University and at USC. She teaches multiple courses at Cal State Fullerton and currently mentors 16 students in her lab. Her research focuses on evolutionary paleoecology, a research area that examines the ecological context of long-term changes in the fossil record. She has spent the last decade establishing and continuing an evolutionary paleoecological project within Central Nevada, which focuses on documenting patterns during Earth's largest mass extinction, approximately 250 million years ago. The title of tonight's talk is Bringing Dark Data into the Light, One Shell at a Time. Please help me welcome Dr. Nicole Bonuso.
Thank you so much for that awesome introduction. And while Julie had mentioned that I do work in Central Nevada, the work that I'm going to present tonight actually focuses in on our work in Southern California. Not a lot of students want to go out to Nevada and do really rugged field work. And so we developed this project to give access to students, a little closer to home projects. So thank you all for coming. I know that you probably could be watching something else right now, but hopefully this presentation will distract you from some of the things you might not be want to watch tonight on TV. So let me start. I want to begin and first kind of give you a bit of background. This work results from a collaborative effort between students at Cal State Fullerton and an organization called EPIC. EPIC is an acronym for Eastern Pacific Invertebrate Communities of the Cenozoic. And it's an organization that helps bring dark data to light. And let me explain what I mean by dark data. Most natural history museums are known for their dinosaur bones and their dioramas, but these institutions are also home to vast collections of fossils used by researchers around the world. Millions of specimens, which is what you're looking at here, these are all museum specimens, help tell a tale of Earth's life history. But the problem is, for decades, the only way to know for sure what was in the collection was by visiting the museum in person and search through the cabinets. And so here are some pictures. These are Cal State Fullerton students. Jolene Dittmar on the left here is searching through, look at these shelves. They're filled with fossils from Orange County and being held in a warehouse in the Cooper Center in Orange County. And then on the right here, these are a bunch of master's and undergrad students from Cal State Fullerton, right? They're looking in all of these cabinets, picking out, they're having fun, right? Looking for specimens that they can work on for their master's and undergrad research projects. But as Jan Vendetti from the LA Natural History Museum put it best, natural history museums are like a library, but a very dysfunctional library. Museums don't have a universal filing method like the Dewey Decimal System. And for decades, many museums simply didn't have the money or the personnel power to properly catalog all the fossils they gather. Thus, the fossils that are in those cabinets are effectively invisible, which is why we often call those invisible fossils dark data. So to solve this problem, 10 natural history museums and academic institutions banded together. They wrote and eventually was awarded a $2 million National Science Foundation, or NSF, grant to help catalog and digitize marine invertebrate fossils along the Eastern Pacific. So essentially, you can see Burke Museum in Seattle, right? This is University of California, in San Francisco. My gosh, I can't even think, sorry. Anyway, I went blank. So all the way from Alaska down to Seattle, through California, and right down here into Southern California, including California Academy of Sciences, LA Natural History Museum, as well as the Cooper Center. And then we have some collaborations on the East Coast as well. So NSF also has been supporting other growing efforts to roughly digitize about 1,600 other collections and trying to bring them online. So once these collections are cataloged, we use our student power, right? This is a student, Cambria Rodriguez, at the LA Natural History Museum facility. She is actually digitizing the then cataloged specimens. And so she props them up. She sometimes has to scrub and clean them before their photo shoot, right? But you can see her there with the lights. You can't see the fossil that she's taking a picture of, but eventually she has a very high quality digital image of that fossil and its identification. Like I said, Cambria is from Cal State Dominguez Hills. I might not have said that, but we also have students from Cal State Fullerton, like Jolene Dittmar, who worked at the Cooper Center digitizing these specimens. So far, through this digital process, 9 institutions, their holdings within the Cenozoic marine invertebrate collection, all the way from California, Oregon, and Washington, right? This is a paper we all published in 2018 that just represents what kind of collections we had before this digitization that EPIC introduced. So what you're looking at here is a series of little yellow marks throughout California. This is the literature-based online data that we had, right? A repository called the Paleobiology Database. Then after EPIC started digitizing their museum collections, if you look on the right here, you can see all these dark red colors, right? We've increased the number of sites by 23-fold in this digital project that we're doing, right? So we've increased the number of Cenozoic marine invertebrate fossils through the work of EPIC so far. So you can imagine when we fully digitize these data, it'll enable paleontologists to make inferences about the patterns and processes of past ecology and evolution changes within the Eastern Pacific realm. This is really particularly important in an age when we have rapid global climate change, right? Because the past is the key to the future, right? We can tell stories from the past and predict what's going to happen in the future. So let's talk about our study. Our study actually focuses on Southern California. And for those of you who don't know, Southern California provides an excellent laboratory for examining past evolutionary and ecological changes because we have these wonderful sedimentary basins, right? So this is a little figure from a paper that's about to go out for publication. And so we have the Southern California coast here, and the dotted line represents the LA basin. And then this little dotted line here represents the San Diego embayment, which is another sedimentary basin. And for those of you who aren't geologists, sedimentary basins are just big buckets, right? And we know that LA is surrounded by mountains. And when the mountains go up, right, all of those rocks that are being pushed up are eroded, and eventually all those sediments get deposited within these basins. So when we layer those sediments on top of one another, it makes a perfect environment to preserve fossils. And that's exactly what's in these basins. So we have this region contains well-preserved and an extensive fossil record, particularly within the last 65 million years. So within this study, we're actually going to zoom in on a very specific time interval. We're going to focus in on the last 4 million years. Right, our data are actually span. We'll talk a little bit about this later, from the late Pliocene through the Holocene. So we focus on the last 4 million years because examining the species patterns from the last 4 million years provides a suitable analog to modern biotic patterns for several reasons, right? So we want to compare what we see in the past to the present and future. And why this last 4 million years is such a great time interval is because first, I'm going to take you back all the way to the late Pliocene. This is a paleogeographic reconstruction of Southern California. And I'm showing you this because about 3 to 2 million years ago, the continents and the ocean basins have nearly reached the present day configuration, right? So if we want to use the past and compare it to the present and the future, we want to have the same sedimentary basins. And so, yes, sea level was definitely higher. If you can see, this little gray outline here represents the present day coast, and these white, light blue areas are the shallow marine environments. And so the LA basin is sitting right here, and that San Diego embayment is down here to the south. And so when we progress just to look at the paleogeographic reconstruction of the area, in the Pleistocene hasn't changed much, right? We still have that nice LA basin and that San Diego area, and then eventually into the present, just to show you, right, that the continents and the ocean basins are pretty much the same, starting from the last 400 million years ago to today. So that's the first reason why we focus in on the last 4 million years. The second reason we focus in on the last 4 million years is because during that time interval, climate shifts from a sustained warming period to a cooling period. So this is research done by someone else, right, by Burke et al. In 2018, and it just shows lots of squiggly lines. We're going to see a lot of squiggly lines tonight. But these squiggly lines represent changes in temperature, in this case, degrees Celsius. So on the vertical axes, we have change in temperature in degrees C, and on the horizontal axis, we have time, right? So deeper time is to the left, right? And today is on the right. And then on the top here, it just gives these geological time units. Some might be used to these time intervals, the Pliocene, Pleistocene, and Holocene. That's what we're going to focus in on. But just we'll talk, hopefully you'll get to know what the Plio, Pleistocene, and Holocene is by the end of this talk. And so, okay, so the whole point of this is to tell you that it's an interesting time interval because it records a climate shift, right? So we can actually see how marine invertebrates respond to climate change in the past. the last reason why we pick the last 400 million years is that 75% of the California province living mollusks, right? Those are the mollusks that are living, that range through from California all the way down to Baja. 75% of the living ones are actually present in the Pleistocene fossil record, right? So the taxa are the same. And so we can make really good inferences on the present and the future with using taxa that are still here today. Okay, so let's talk about the main research goal of this. Actually, I'm not going to get to the main research goal. I got ahead of myself. Sorry about that. I want to talk about, we talked about why the last 4 million years. We talked about why Southern California. Our study actually gets a little bit more specific because we focus in on oyster communities. And so we're interested, this study is interested in reconstructing oyster communities through time. And the reason why is because along the North American West Coast, from Alaska to central Baja, there's only one native oyster species. And this is a picture of the Olympia oyster. The scientific name is Australurida. Within the last century, it was widely distributed in the bays and estuaries, including in California. And it was exploited as a food resource by California Native Americans. But in the early 1900s, some combination of overharvesting, pollution, habitat loss led to significant declines throughout the species range. So oyster beds are now absent in California estuaries. There are like really very few low diversity populations exist, but in general, oyster communities are completely gone from our coasts. So currently, biologists like Dr. Danielle Zackrell from Cal State Fullerton work to create diverse native oyster beds within Southern California. And so these are images of all the volunteers. They lay out a substrate within these estuaries so oyster larvae can attach to it and oyster reefs can build. But the goal, right? So they lay these down, they hope they actually do have oysters growing in the bay. But the goal of their restoration is to restore ecological niche that is ecologically functional and resilient to changing environmental conditions. So Danielle and I are friends, colleagues, friends, and we just got talking when she was telling me about her research, and I asked her, I was like, well, what's your baseline? What do you, what's your goal of restoration? What are you aiming? What's your, what makes a resilient community structure, right? And so she's like, that's a really good question. And then we both started talking and we realized that, there's no quantitative data at all describing oyster abundance and ecological patterns prior to the oyster's modern decline. None exist within Southern California. And so this opened up a great opportunity to look at the fossil record and see what oyster communities look like in the fossil record. So we argue that examining past oyster communities from the late Pliocene through the Holocene not only will help establish pre-human oyster community baselines, but we can also provide some insight into how oyster communities respond to changing climate. So it's like a double whammy here, right? Win-win. So now I'll get to my our main research goal. So our main research goal is to reconstruct species occurrence patterns by determining relationships among taxa through time. And the way that we do that is we look at the fossil record and we document which taxa or species vary through time. But we also want to take a look at how these taxa or species vary along the coast of Southern California within those particular time intervals. So what do our data and analyses look like? So I've been talking about EPIC and how they digitized all of these fossils. And so we absolutely use epic data. But I want to describe to you what epic data actually is. So it's a little pixelated, but these are actual boxes of fossils that have been identified and counted, right? So this little box here represents one genus and species of a fossil, and all of the individuals in there, right? We count them and get an abundance of that specific species. So the EPIC database contains fossil abundance data that are all counted and identified from one specific locality or area. And so it's so unfortunate that within Southern California, the collection sites are either you can't access them or they're completely gone, right? They literally ripped up sites within Long Beach, right? Because of urbanization. And so all the fossil localities are gone. So the data within the museum is all we have. And so talk about dark data, right? So this is fantastic that we have access to these types of data. So in addition to utilizing EPIC data, we actually had some collecting of our own. Here's Jolene again. We had a handful of undergraduate students working. They actually screen washed and counted identified fossils from 2 localities. First, we have a locality in the Holocene. Kelly Vreeland worked on her master's thesis, and one part of her master's thesis was counting and identifying oyster beds within Newport Bay in the Holocene, as I mentioned. And then here's Jolene. She's actually looking at, she's working on sieving and counting and identifying late Pleistocene Palos Verdes sand samples from San Pedro. So we have a combination. We collected some data, but the bulk of our data come from EPIC. So let's look at time again. When we took all that data, we searched within that database specifically, calling up all the fossil localities that had the presence of oysters, right, within the last 3.6 million years, so roughly 4 million years. So we have data from the late Pliocene, the middle Pleistocene, the late Pleistocene, and the Holocene. Now you all must be wondering, well, what the heck happened to the early, why don't we have data from the early Pleistocene? Within the Southern California region, we don't have sediments from that time interval. They've all been eroded away. So no sediments, no fossils. So once we actually, combined EPIC data with the samples that we collected, we broke our data down into these 4 time bins. We then used statistical analysis to explore species patterns or species abundance dominance patterns through time. And we specifically used a statistical technique that identifies significant groups of species which coexisted together, right? So we're looking at which organisms are coherent, right? Which ones are occurring together. Okay, so let's look at some results. How do species change through the time interval of this study? I'm going to do some explaining here. I told you you're going to see a lot of squiggles. First, what I'm going to do is I'm going to show you the species patterns through time. So this figure is showing you data the data within the late Pliocene coherent groups, right? So again, here's the late Pliocene. So now here are the bunch of squiggles. So let's walk through this figure a little bit. On the horizontal axis, we have the Pliocene on the bottom right. And as you progress to the left in the horizontal axes, you get to the Holocene or present. So time is on the horizontal axes. Where on the vertical axes, we have abundance, percentage of abundance through percentage abundance from 0 to 100 at the top. So now you see all of these squiggly lines. And they're all colored, right? So that's fantastic. So each color actually represents a specific species. And so what the great wonders of statistics does is help us group the taxa that co-occur. And so this top group here is group B, right? So there are a bunch of different colors. Each color represents a different taxa, and it shows the abundance of that taxa. And so, right, if you look at this little green line, right, that's a specific taxa, and it, has about 90% abundance within this sample, right, within the Pleistocene. So that's how you read the graph. And so I want to just focus in on the late Pliocene data. I'm trying not to confuse my P's. That's why I'm speaking so slowly. So everything highlighted here are data within the late Pliocene. And what you can see here is that group B, the top group, right, it has several really abundant taxa. One taxa that I pulled out here is Dendrostra. That is the abundant oyster within this time period. So what you can see is that we have multiple taxa, really, really abundant, and they're co-occurring within this group B, within the Pliocene. And so just give you a couple pictures. I didn't give you the pictures of all of them, but there's Dendrostra. That's our dominant oyster. And then we have some, I call them cute little gastropods, and a few different bivalves that are really abundant and are coherent groups. They co-occur within group B. But then as we march down this diagram here, you can see group C taxa. They're not really abundant. They're only one taxa, one species is actually present within group C. So not really abundant. Group C is not abundant at all within the late Pliocene. And then as we move down to group E, we have one highly abundant, I would call that a rare species, abundance pattern there, and then a few species in group B. So the take-home message here is that the top, group B, these taxa are the most abundant taxa occurring within the late Pliocene. Specifically, as I mentioned, Dendrostra is that dominant oyster. So now that I've spent a little time explaining how this figure works, we're going to just keep marching on through time. So what does the mid-Pleistocene look like? If we highlight that area, right, we can see group B is non-existent, doesn't exist within the middle Pleistocene, right? Pretty much doesn't exist through the Pleistocene or the Holocene, right? Just flat lines through the rest of time. But as we move down to group C pops up, and we start to see abundance patterns within the taxa that are co-occurring there. Within group C, we have the dominant oyster, Ostrea conchophilia lurida, right? So we have a new oyster that actually appears and becomes abundant in the mid-Pleistocene. And then again, here are the other two taxa that are abundant during that time, two bivalves right there. So as we march down these groups, right, group E, there's only one taxon, that is abundant within that time period, as well as group G. So the take-home message here is that within the mid-Pleistocene, right, we have a really, group C has the most dominant co-occurring taxa, including a new oyster, Ostrea conchophilia lurida. So as we progress through the late Pleistocene, we can see group C still continues on, right? As I mentioned, group B, pretty much non-existent. So group C continues, the same group that we saw in the middle Pleistocene, but then we actually get another group of taxa becoming abundant and dominant, right? A new gastropod and two new bivalves become abundant. And then group G is kind of a lame, a lame group. It just has a couple of taxa being dominant in one sample. And so we interpret that as being not a really robust signal of dominance. So take home message, in the late Pleistocene, we have group C and group E taxa being very dominant. As we progress into the Holocene, right, this little sliver of data, we can see, right, group B is not there. Group C is still dominant. Same dominant community that we saw in the middle Pleistocene, late Pleistocene, and now in the Holocene. Group E was there, but very low abundance. Sorry, I clicked too fast. So in summary, what do we see through time? Here's a close-up picture of Austria Conchophilia lurida and some other Palos Verdes sand specimens. So in sum, what do we see through time? Within our data, a regional extinction occurs at the Pliocene boundary. We see it not only within the dominant oyster, but 9 other, so 10 total species out of the 30 most abundant species go extinct, including the dominant oyster. Oyster communities start to recover and restructure during the middle Pleistocene, and we start to see a new dominant oyster take over. And then that new community that restructured within the mid-Pleistocene continues and remains stable through the Holocene. So that's our summary of dominance through time. How do species change spatially? Let's look at that data. So we're going to look at some more squiggly lines, and we're going to look at them through their different time intervals. And so here we're going to look at the late Pleistocene and look at the spatial patterns of these coherent groups through space. And what I mean by space is, right, so again, here is our little cartoon of Southern California coast, right? And all of these little symbols here represent the samples that we looked at and where. So we're going to actually look at how those coherent groups change or remain stable along the coast of Southern California. Okay, so again, more of these squiggly figures, but I've changed the horizontal axis on you, right? Because we're looking at spatial patterns, I've changed the axes, the horizontal axis to represent space. So the Los Angeles County samples are on the left, Orange County and San Diego. So the horizontal axis is actually sorted to represent the northwestern samples going all the way down to the southern southeast last sample right here in San Diego. And so what you can see within this data is that there are a lot of co-occurring taxa. But when we look at the, when we look at the majority of the data, so again, we have a couple of spikes, but the majority of those taxa are varying in abundance across all the different counties, right? We can see, like, if you just look the red line, or well, maybe you can see the red line, but the red line here represents a species. It's high in LA and Orange County. It gets a little bit low in San Diego, but then it peaks up again in San Diego. So that means that species is actually ranging through all 3 counties all along the southern coast. Again, I picked out what the dominant oyster is doing, and you can see Dendrostra is low in abundance in LA and in Orange, and then peaks a bit in San Diego. But again, it's still ranging through the Southern California coast here. So that's one group. Another group, mostly pectins, these bivalves here. Again, if you look at the majority of abundances, they're ranging through the different counties here. And so we interpret the community within the late Pliocene to be a loose I'm messing up my words. It's a loose community where the individuals are migrating along the Southern California coast. Okay, so let's look at the middle Pleistocene. When we look at our middle Pleistocene data, we actually get significant turnover within our spatial sampling patterns here. So what do I mean by that? Again, along the horizontal axis here, we have space. We have LA County and San Diego County. Unfortunately, we didn't have Orange County samples for the middle Pleistocene, but what you can see here is we have really, really high abundances for group B taxa. Here are two examples of some of the taxa within group B. Really high abundances within the northwestern samples of LA County. And as you progress further down within the coast, right, group A becomes really, really abundant. Group A actually has the dominant oyster, Osteoconchophila lurida, as well as other taxa, but really, really dominant within the more southern Los Angeles County samples. And then when we move into San Diego, a whole different group pops up and becomes dominant, right? More pectins and another bivalve within that group C. So we interpret this pattern of community within these coherent groups, right? This represents a community that is isolated, right? So migration is low between the different communities within the Southern California coast, right? And you can see those groups, right? There's different dominance, different samples along the Southern California coast. Okay, so let's move on to the late Pliocene. Within the late Pliocene, oh my God, I said late Pliocene. Within the late Pleistocene, sorry, get my P's mixed up. We see a similar pattern that we saw in the late Pliocene. So late Pleistocene spatial coherent groups. So when we look at the majority of abundance patterns within the late Pleistocene, we can see taxa, right? Their dominance, their abundance is fluctuating through waxing and waning in the different samples along the coast, right? LA, Orange, and now San Diego. So this little green line represents the dominant oyster, and we can see it ranges right through, as well as other taxa within group D. Another significant coherent group is group A, again, pectins, right? They like to hang out together. And so we can see the majority of abundances within group A are ranging through or waxing and waning through time. So just like the late Pliocene, we interpret these communities as loosely structured, where individuals are actually migrating along our coast. Okay, so let's look at the Holocene. Holocene looks a lot like the late Pleistocene, right? We only have one significant coherent group here, but the taxa are varying in abundance between the two different counties. We don't have San Diego samples here, but between LA and Orange County, right, we can see that group is varying in abundance spatially. So again, we interpret this as being a loosely structured community where individuals are migrating along the coast. Okay, so let's sum up the patterns we see through space. So spatially along the Southern California coast, we see that the late Pliocene communities reveal little turnover between Southern California localities. So the majority of the species are migrating between localities, representing this individualistic loose community structure. Whereas we get to the mid-Pleistocene, communities reveal significant turnover between Southern California localities. And instead of migrating, right, these communities are now isolated. They're within specific areas along the Southern California coast. And then once we get back into the late Pleistocene and the Holocene, that loosely structured community that is that we saw in the late Pliocene returns, right? So the taxa begin to migrate along the coast again within the late Pliocene and um, into the Holocene. So those are our results for this research project, but I want to talk a little bit about what might cause the patterns we see. So here, I promised you lots of squiggly lines. And again, this is the figure that you saw in the beginning of the talk by Burke et al. 2018. Just to review, horizontal axis is time, right? Deeper time is on the left, the present is on the right. And what we're looking at is changes in temperature, degrees Celsius. And so I want to point something out to you, something that I didn't talk about last time. I mentioned that there's a warming period, we get into cooling period as we progress into the Holocene, but I just want to point out this little gray line here. This represents the initiation or onset of the Northern Hemisphere ice sheets, right? Glaciers in the Northern Hemisphere. So previous research, I got ahead of myself. Sorry, I do want to focus in on this Northern Hemisphere ice sheet onset, but our data, right, we see this shift right, we see a local extinction and we do see a change within oysters, dominant oyster, right at that Plio-Pleistocene boundary. And it just so happens to coincide with the onset of Northern Hemisphere, the growth of that ice sheet. So previous research actually documents other Plio-Pleistocene fossils along the Eastern Pacific. And two different papers report that the magnitude and the rate of temperature change between 1 million year periods correlates significantly with extinction at the Plio-Pleistocene boundary. So we conclude, we suggest that most likely the cause of the regional extinction that we see is probably due to this onset of the Northern Hemisphere glaciation, right? There's not only does the magnitude of temperature change, but the rate changes as well, right? So the magnitude and rate of temperature change, soon as we start turning on that Northern Hemisphere ice sheet, right? It changes so fast that organisms go extinct. So we see that in our data and other people have seen that. And so we associate that change with climate change. So what about some other research that I want to present tonight is that we know, as I mentioned, that oyster communities reestablish themselves in the middle Pleistocene. And I said that a new dominant oyster, emerges. But I also show, right, I also showed that in the middle Pleistocene, right, we have those localized communities emerging along our coast. So one of the reasons why we might have this tight, isolated community structure might have to do with what's going on climatically. So the mid-Pleistocene, right, which is right here in the squiggly line, climate researchers Lisecki and Ramo 2005 and 2007, and other paleoclimatologists document this mid-Pleistocene that actually marks transition between short, mild, 41,000 year cycles to longer, more variable 100,000 year glacial interglacial cycles. And you can actually see that in the data, right? You have these shorter term variation, and then all of a sudden you start seeing these 100,000 year glacial interglacial cycles. And so we suspect that perhaps the Southern California marine communities are responding to the water mass differences associated with the mid-Pleistocene transition that occurs during this time interval. Okay, so climate, we think, affects these Southern California oyster communities. So what does this mean for oyster community, their future? So other researchers that have looked at Eastern Pacific localities, they also reveal this loose individualistic community structure. And the way they describe these communities with this loose structure and how they migrate along the coast. Um, and I just want to explain that if you have these loose community structures, um, the since they're not tightly structured, the individuals, the species can migrate along the coast, and they're able to actually track shifting environments, right? So throughout the Pleistocene and the Holocene, as we get these fluctuations with glacial and interglacial cycles, these taxa are able to keep up with those changes. And but as we've seen in the late Pliocene, right, if the rate of change, sea surface change, changes really significantly, um, right, that might affect oyster communities negatively. So um, most likely, right, we see this stability within the late Pleistocene and Holocene, right? Most likely, the sea surface temperature changes during this time didn't necessarily exceed the rate of change occurring at the Pliocene boundary. Thus, so those communities remain stable. So what happens? So I actually think I shouldn't have clicked back, but here, let's click. Let's just stick on this click right here. I think this is good news for us, right? I hate talking about communities and changing and extinction and climate because it always seems like we end on a bad note. But the good news here is that the late Pleistocene and Holocene, those communities are actually pretty stable. They're able to keep up with the glacial interglacial cycles, right? The sort of bad news here is we know that as anthropogenic climate change, right, as right, is that rate of anthropogenic climate change is really much higher, a higher rate than we've seen in the past. And so I just hate to say it, but if we increase the rate of sea surface temperature, right, if we continue to increase the rate of sea surface temperature, it could cause oyster communities to restructure the same way they did in the Pliocene boundary. So hopefully we can moderate and keep that rate of sea surface temperature to not increase so much, and we can keep our oyster communities within Southern California. Okay, so that's it for my talk. The very, very last thing that I want to say is thank you. Actually, that's not the very last thing I want to say. Thank you for listening. But more importantly, I want to acknowledge a whole bunch of people that made this research possible. Starting with Kelly Vreeland. She was an undergrad and a master's student within our lab, and specifically for her master's at Cal State Fullerton, she documented oysters within the last 65 million years. I mentioned her, her data in this project was used, just her Holocene data. Jolene Dittmar, she actually completed a Bachelor's of Arts, did her undergrad thesis that the late Pleistocene, contributed a bunch of data to this research, but more specifically, single-handedly, Jolene digitized all the fossils that she was able to, because there were some time constraints on the grant at the Cooper Center. So she did all the digital images for us at the Cooper Center. Dr. Austin Hendy at the LA Natural History Museum. Lots of chats I've had with Austin, and he definitely helped me download and manipulate the data for this project. Dr. Danielle Zackrell, she is in the biology department at Cal State Fullerton. We've chatted all kinds about oysters, but she's significantly increased my knowledge of oysters, as well as contributed to the manuscript that we're about to publish. Others that are not pictured here, Brian Rue, Julian, they also did a lot of screenwashing, identifying and counting. Sean at now at LA Natural History Museum, helped us with all of those gastropod identifications. Thank you so much. Crystal Cortez, she was helping us with specimens at the Cooper Center. Dr. Parham is in our geology department at Cal State Fullerton. He actually funded a lot of the work that Jolene did with that digital processing. So thank you for NSF for funding, Cal State Fullerton for funding, and obviously for EPIC and all of the efforts that are put forth for that project. So thank you again for listening, and I would love to hear any comments, questions. Let's chat.
Awesome. You don't get applause because
I know.
Quiet, so I'll give you some applause. That was awesome. Thanks, Nicole. That was a great talk. Yeah. So there are some questions coming in, and it's just before 8 o'clock, so we definitely have some time. The first question, yeah, there's some people saying they're clapping virtually,
nice.
So everyone's clapping. One of the first questions, let's see, it came in early on the chat from Marion. She said that she's pointing out that you're talking about scallops, clams, and oysters, and even snails. I saw some gastropods in there too. But she's wondering if you ever come across any abalone fossils, and are there any abalone fossils?
So within our data, no, but they're probably living in no, I mean, no, not within our data. They're not there. But that doesn't, these are just fossil collections. The abalone, those are the big ones that Jolene was holding, right? Like, so that's, sorry, that's this one. I just want to make sure we're talking about the same thing. Where is she? She's all the way back here. That's this bad boy, right? Well, that's, a very large one, but you're talking about this specimen, not the, right? She's actually holding the shell.
There it is. Oh yeah, that's right. That's an abalone. Yeah, that's a big abalone.
Yeah, that's not from our, yeah, there's a huge barnacle. So it's probably from the Eocene, I'm guessing, because they're huge fossils in the Eocene. But no, we actually don't see them in our data at all.
Okay. Let's see. Brett has a question. What do you think helps to maintain the stability of oysters in the Pliocene era or warmer climates?
Yeah, so I think that these taxa aren't dependent, these species aren't dependent on one another. And so there's a lot of research that shows, so the reason why these species have very large latitudinal ranges, so they can withstand big changes in temperatures. That's just who they are, these taxa. And so they're able to actually migrate along the coast. And so because of these great geographic ranges, right, they can keep up with fluctuations in sea surface temperature changes. There's actually a lot of research. There are a couple, not a lot. So there are 2 papers that talk about the geographic range of these specimens within the last 4 million years, and talk about how it's easier to, let me get this straight. So it's easier to migrate to the north than from the south. So it's easier for the northern species to come south, not the southern species to come north. But that has to do with the differences in water temperature. Anyway, so yeah, it has to do with the fact that these taxa have long-ranging geographic ranges.
Okay. Cynthia is asking, she's making a comment that she's heard that a lot of scientists and people tell everyone not to take shells from the ocean, and because other animals can use them for homes, and the oyster shells can let other oysters stick to them. But she's wondering if there would be any reason to See, not that I want to destroy data or take them from museums, but would throwing these shells back in the ocean help anything?
Yeah, so this is why I clicked back to
Mhm.
Dr. Zackrell's images, because what they do is they actually, see what they're carrying right there? They're plastic buckets, and those are all dead oysters. So what they do is they put these burlap sack nets out in these estuaries, and then they pour dead shells onto them. So in the waters along Southern California, the larvae are actually floating around in the water. And so they're looking for something to attach. So, yeah, go throw some dead shells in the ocean, right? That would be helpful. Just don't throw the plastic or, right, anything like that. It's so sad to see how much stuff is along the coast. But yeah, so those dead shells are not only habitats for, um, I was thinking about those crabs. I can't think of their names right now, but yes. So the dead shells can help restore oyster reefs, but also provide, other resources for other organisms.
Okay. Let's see. Here's a question from someone that's anonymous. Is there data about how deep or in what habitat these oysters lived?
So are there data? Sorry, I just want to repeat it to make sure I heard it correctly. So are there data on the depth at which these oysters live?
Yeah, and what habitat they lived in. Like what was the habitat like and how deep were they? Is there information on that?
Yeah, so we're talking about shallow marine. So not where, not where the ocean is coming up and crashing onto the shore. We're talking about, so what you're looking at here in these are estuaries. So these are very shallow water, uh calm, right? You don't see like big crashing waves, bays and estuaries. And so that's where these oysters would be living.
Okay. Dawn has a question. How was Julian able to participate as a high school student? Are there opportunities for other high school students?
Yeah, there's always opportunity. We could always find opportunities for high school students. Julian was just so super excited about paleontology. And so there's just extra paperwork you need to do. As a faculty member, we're already certified to, there's certain regulations you have to have when you're have minors in your lab, but we're already certified to do that. And so you just have to fill out some paperwork, and Julian can only work with me and not other members within the lab. Like I have to be present there legally when he was working there. So it's not that big of a deal. And we had for this project a lot of samples, and so it was it wasn't easy, but that's what I love about this project. It's very accessible for all students. And so, yeah, and so it's just paperwork and whoever is interested. So if you have high school students, definitely, I don't know if you have my contact information. Is that available?
I don't know that you can
But
you
my
can
name,
yeah, say it.
my last name is very unique. And so if you just Google my last name and CSUF, it'll come right up.
Cool. We can also maybe put it in the chat later.
Yeah.
But yeah, it's probably easy to Google you and find your email address.
Yeah.
Okay, another question from George. This database must have been key in your research. Is the digitizing of fossil samples an ongoing project for all fossils in the efforts to provide a database for all researchers, scientists, museums, or the public to access? If so, is there a website for public access? And PS, go Titans.
Nice. Love Titans. Um, of course, right? Okay, so I'm part of two of these databases. So the one that I presented, this literature database. So I just want to explain a little bit about it. So what we've been doing for at least 2 decades is that we mine peer-reviewed literature, right? And we see their fossil counts and numbers, and we enter them in this database. That's called the paleobiology database. Again, you probably can just search that, and a website would pop up, and there's ways to access that. Oh, anyway, I'm not gonna go to the website right now because I don't want to ruin what we have up here. But you could search that database. Um, the EPIC data is only focused. So the literature database, the paleobiology database, is national, right? So there are fossils all over the United States. We haven't really branched out internationally. So not all the fossils, not yet. There's a lot of fossils, but lots of data is in that paleobiology database. The museum collections, the EPIC collections, our project is just focused on the Eastern Pacific. So California, Oregon, Washington, Alaska. And so we're not even anywhere near done digitizing the fossil collections in those museums. And so, like I said, the projects that I'm associated with, right, those are just various, Eastern Pacific related and then literature related. But then other museums have also asked for funding with from the National Science Foundation, and they're doing their own database. One thing that you might want to search for is our data repository is called iDigBio. And the data are there, but they're not readily, it's not in a good format for the public just yet. Whereas the paleobiology database absolutely is ready for the public to view, and you can just search through that database very easily. I hope that answers the question.
Yeah, I think so. Okay. Craig wants to know, were there similar studies to yours in areas to the north? Can you talk a little bit about what they found?
So similar studies to mine, definitely not. The studies that I had mentioned that are talking about these loosely structured communities. So I mentioned in this figure here, sorry, I have to just get rid of a couple things on my screen to see what I can. So I mentioned that there are studies along the coast here. Most of the data. So Jablonski did a lot of work in the Eastern Pacific, mostly in California. This Edie paper looks at a whole bunch of different time intervals and talks a little bit about the Pleistocene. And so these studies mostly are just looking at diversity and how diversity changes through time. My study, and I don't really know of any studies looking at not only the diversity, right? Because we talked about how different oysters change, right? So there's a taxonomic change, but there's also an abundance and dominance change that's going on. There aren't a lot of diversity studies. And what's different about our study is that we look at dominance, right? So the abundance of taxa and how they vary through time. And so those studies are a little bit different. So yeah. So I don't know of any studies like the one that we're doing. And I know that from the biology side of things, they're, as particularly in Southern California, they're waiting. They want to know what's going on. They want to see these results because nothing has been done in Southern California in terms of oyster communities in the past.
We'd like to thank Nicole. We'd like to thank Friends of Cabruman Aquarium for their support and the City of Los Angeles. And for more information, everything's going to be posted on our website. And like I said, this is recorded and we will post it soon. So hopefully we'll see you guys again on December 4th. And hopefully I will have an update then about the next speakers in 2021. The next one will be February. We'll take January off and the next one will be February. So hopefully I can announce to you who that will be. And thank you again for joining us. Stay safe and good night.